The next talk is by David Enard from Stanford.
Hi everyone, I'm David Enard. I'm a postdoc in the Petrov Lab, and today I'm going to talk about how viruses drive adaptation in human and other mammals, and how signals of adaptation in those hosts can be used to study ancient viral epidemics. Viruses are known to be one of the strongest selective pressures, and the nice thing about them is that we actually have some historical measurements that we can use to back it up. So for instance, if you go back to London in the 18th century, you can see from historical records that smallpox virus alone explained 10% of all deaths. So it's a really, really strong selective pressure. And in line with this, extreme signals of adaptive evolution have been studied in specialized antiviral proteins. But those antiviral proteins that are very, very specialized immune proteins do not represent all the proteins that viruses interact with in their hosts. And in fact, there are many, many thousands of proteins that viruses interact with in human and other mammals. And those are very diverse function and do very, very different things in their host. So our first question, if you wanted to study adaptation in those proteins, was to really wonder how many of them there are. So what we did first was to go through months of manual curation of the low-throughput literature in virology, and from this we could find 1,900 virus-interacting proteins. We call them VIPs, and it's proteins that interact physically with viruses. So in addition to these 1,900 VIPs, we also used 2,600 VIPs that were detected with high-throughput methods by virologists. So in total, that's about 20% of the human proteome that is currently known to interact with viruses, which is pretty enormous. And that's just to give an idea of the sampling of different viruses that we have so far. So what we did was, once we had those 4,500 VIPs, we compared them to all non-VIPs, meaning all proteins that do not interact with viruses, that have not been shown to interact with viruses. And we compared adaptation in those two sets at these different evolutionary timescales. So first we looked at adaptive amino acid changes in mammals, then more specifically in the human branch. And then we focused more specifically on recent adaptation in human, looking at adaptive integrations between Neanderthal and modern humans, and then focusing really on the most recent signals of adaptation that you can possibly detect by looking at haplotypes in extant human populations. So first, looking at adaptive amino acid changes. I will go quick on this because it's been published already. If you're interested, you can look at this paper. What we did in this study was to focus on 1,300 VIPs that are manually curated and have orthologs in all the mammals that have been sequenced so far. And we compared those VIPs with 8,600 non-VIPs. And what we found for those is that in mammals, you have about 2 times more adaptation in VIPs compared to non-VIPs. And this excess corresponds to about 80,000 independent adaptive events. Now, if you focus on human, what you find is that there were about 500 adaptive events in VIPs that we could estimate versus 1,300 adaptive events in non-VIPs. So although VIPs represent about 13% of the proteins that are included in this study, they account for a very substantial 30% of adaptation in human evolution. So that really means that viruses have had a very, very, very quantitatively important impact on adaptation in human proteins. Given these results, it was pretty straightforward to hypothesize that we might also be able to detect a substantial impact of viruses on recent human adaptation. And what we did first was to look at adaptive integrations between Neanderthals and modern humans, because if you think about it, if Neanderthal and modern humans interbred, they probably exchanged a bunch of viruses that they had not experienced before when they came into contact. To look at adaptive integrations, it's useful to first come back to a more classic example of hitchhiking, which is hitchhiking when you have a new adaptive mutation. Here in red, you have several haplotypes in a population. A new mutation happens, and after a number of generations, you can see that the haplotype that carries the new adaptive mutation has also increased in frequency. And the way you can detect adaptation in that case is by detecting that the haplotype is unusually large and at an unusually large frequency. It's the same that happens when you have an integration from another species that jumps into your population. In the process of hitchhiking, what you're going to have is an integrase segment that is at unusually large frequency and unusually large size. So this is what we looked at for Neanderthal using the data from this paper. And what we did more specifically was comparing VIPs with carefully chosen genomic control using a bootstrap approach. And what we asked was if there was an excess of adaptive integrations in VIPs compared to this control. So I'm going to show it plots that are structured this way. If you go down on the plot, you go from increasingly large, from small segments of integration to large segments. And if you go from left to right, you go from low frequency to high frequency. And the lower right corner is the region of the heat plots I'm going to show that are more likely to have adaptive integration. The color code is blue, you have less integrations than expected. Red is you have more integrations than expected. So we first looked at integrations from Neanderthal to Asian populations. And we found some limited evidence first here of an excess of adaptive integration in Asian populations. But what we know from adaptive integration is that it's much easier for integration to happen in high recombination regions of a genome. So what we did then was to repeat the same analysis, but looking at increasingly higher regions of the human genome. And when we do this, this is what we get. So the signal of an excess of adaptive integration in VIPs becomes much clearer and stronger. Meaning that probably viruses have driven several adaptive integrations from Neanderthal to Asians. When we do the same in Europe, we actually find an even stronger signal of adaptive integrations from Neanderthal to Europeans driven by viruses. Now that was going from Neanderthals to modern humans. If we now go from modern humans to Neanderthals, using the results from this recent paper, we find the same signal. So this time, the excess is not coded as colors anymore. It's coded as the y-axis. The reason is that there is no frequency to take into account here, since integrations from modern humans to Neanderthals were just seen with one Neanderthal individual in this study. And again, we see that the excess of integration increases as you look at increasingly large integrase segments, which is a sign of an adaptive integration. So that was for integrations between Neanderthal and modern humans. Now, if we look at the most recent signals of recent adaptation that we can look at in human, looking at adaptive haplotypes, it covers about 10,000 years of past human evolution. This time we looked at new adaptive mutations driving haplotypes to a large frequency, and especially large haplotypes being driven to a high frequency. To do this, we used the IHS statistic, which is a pretty well-known statistic to study partial sweeps. And when we measured IHS on the 1,000 genomes phase 3, we ranked all the genes in all the human populations by how strong the IHS signal was. And what we could find was that the top ranking IHS candidates in human populations are very, very, very strongly enriched for VIPs. So it really looks like VIPs have driven a very significant proportion of those recent strong adaptive events. And what we asked then is that if we have such an excess, that probably means we can detect specific viruses having driven adaptation in specific populations. And when we did this, we found that 2 viruses seem to have had an impact on specific populations recently. So HIV in Europe and Kaposi sarcoma herpes virus in Africa and in the Americas. So obviously, I'm not talking here about those viruses having driven these signals of adaptation, but those viruses just act as proxies for more ancient viruses that are related to those extant viruses. If we focus more specifically on the signal related to HIV, we could confirm that we have a signal by using carefully chosen controls. And when we looked at the excess of adaptation, we see that it's indeed strongest in Europe and weakest in Africa, with all the other populations being between these 2 populations. So it really looks like some ancient HIV-like virus has driven signals of adaptation, most recently in Europe, and perhaps further back in time in other populations. So this result basically shows that there is enough adaptation, and there is enough adaptation driven by viruses in human population to make it possible to study ancient viral epidemics from the point of view of the signals of adaptation in the host. And if we could do that with other species, that means we could basically learn about past viral epidemics in many, many different species. With this, I would like to thank Dimitri and the whole Petrov lab and our collaborators on this project. Thanks.
We have time for questions.
So do you have a sense of how biased your search for viruses is based on the literature you put in your search? Because, for instance, HIV, I imagine there's lots of literature about this virus.
Yeah, there's a strong bias that's related to the effort of research that is conducted on different viruses. So definitely, not too surprisingly, HIV and influenza virus are the ones that have by far the most VIPs. But basically what that means is that for the other viruses, there are still many VIPs that have not been discovered yet. So we're still probably pretty far from knowing all the proteins in the host that interact with viruses. Yeah.
Do you test it for signals of balancing selection?
Yeah, so the question was, did I test if there was an excess of balancing selection in VIPs? We have just started to do it, so it's still pretty unclear.
So to follow up on the previous question, I mean, if you had enough data, wouldn't every gene in the human genome be associated with a virus?
Probably maybe not every gene, because they still tend to interact with proteins that are like more conserved at the protein level, and they tend to interact with proteins that are expressed in specific tissues. But
right. So I guess to get back at this, you have 1,100 proteins that HIV interacts with, but
yep.
there's got to be, that seems like you're got a binary on or off, interaction or not, when you imagine that's a much more quantitative
Oh, sorry,
trait.
can you repeat that? again?
It's a much more quantitative trait than does it interact or not.
Sorry, I don't.
It's not just on and off, it's just the level of interaction.
Right.
Oh, yeah.
So HIV interacts with 1,100 proteins according to here, but there must be some where the interactions are very minor. And where
Oh,
there's
yeah,
very-
okay, I see what, yeah. Yeah, that's true that what we did is kind of black and white at this point, but it's a future direction of research to include more solar signals. The nice thing, though about classifying things this way is that it's a very objective criterion that we are using. Either you are known to interact physically or you're not. So it's a good starting point to see if there's any signal to detect in the first place.
Thank you.
Right. Thank you very much.